Hello, everyone. Sorry we are late. Good evening, good afternoon, good morning. I'm Ali. I am with the Lilypad Project, and I'm joined here, of course, with my awesome co-host, Kartika, again. Hello, Kartika. How are you today?
I'm really good, thank you. Great to see
Awesome.
everyone.
Yes. Thanks for joining us. We're sorry we're late today. Unfortunately, our main star today, Niklas from LabDAO, is AFK. We think he must have just got the time wrong because he was super excited to join us here today. But all good. We're just going to talk for a little while about some of the saving of data that Ben from Protocol Labs, who is here with us as well, and I will invite him to the stage. Hi, Ben, aka Wings. Who's a
Hello, how's it going?
fellow Australian. Sorry, I interrupted the accent reveal there.
I'm told it's more of an American accent than an Australian one, which is funny. But
Yeah, you do have a bit of an odd, it's not super Aussie accent. It's a good point. But anyway, fellow Australian over on the other side of Australia, Western Australia. If that's still part of Australia, that's an in joke.
we're seceding next month.
This is what we always say about West Australia. It's so far away that it's really not part of Australia anymore. They're leaving. They're 3 hours behind as well. It's crazy, the time difference. Anyway, all good. Moving on here. Ben, I'd love to talk to you about this data save that you did recently. So I hear you saved about 38 terabytes of open science data from not being available. Oh my gosh, just before we go to that, guess who's just turned up?
Yes.
All our calls.
It worked.
Hey.
Niklas!
Sorry for being late, everyone. It's so good to see you all. Hey.
Let me tell you, it's very good to see you too, Niklas, because I didn't know what we were going to do for an hour. Great to have you here. Niklas is the founder of LabDAO. We're very, very excited to have him here today. We were just talking about how you and Ben actually saved like 38 terabytes of open science data from, basically not being available to any scientists anymore. And I've got it here on IPFS. Ben's shared it with me, but maybe Ben, you could kick us off and tell me what this is all about and why you saved it, why it was a mission, how this all came about. I want to hear the whole story.
Sure. So I'll let Niklas tell some of it, but like from my perspective, so I work at Protocol Labs. I would describe myself as like an infrastructure engineer. Solutions architect is like my title. I have been running infrastructure for a project called FDI, which was hosted at Protocol Labs, and I was just browsing Twitter, X, whatever we call it now, and I saw basically a call for help coming out that was saying like, "Hey, this like resource by Meta AI is shutting down, or the team that was behind this resource is shutting down. We have all this data that's now endangered. Does anyone have like storage that they can that they might have spare that they can like use to mirror this?" And my first thought was, I have a petabyte sitting at a friend's place worth of storage. Technically 1.3 now, if anyone's counting. And then also we have 1.8 petabytes of storage at FDI. And so I thought, okay, well, we have storage, we have bandwidth. Why don't we mirror it? And PL and Protocol Labs is like particularly well suited to this sort of rescue operation, just because of like our expertise in IPFS and the open web and Filecoin and all kinds of other things. So I saw that there was a call for help there, and I started like frantically pinging everyone that I could involved in the thread. And then also Niklas, I reached out on Slack to various parts of Filecoin Slack, asking for help as well. And so I was like pinging everyone, going like, where is this data set? I want to start preserving it as soon as possible, because as soon as I saw the call for help, I just like, the mission started, right? So eventually I worked out that it was all like scripts on GitHub and you could just like start downloading it. And so without really asking anyone, I just started pulling it down as fast as I could. I think we peaked at 7 gigabit per second. So someone, I'm sure if someone was like watching the traffic logs at Meta, they probably wouldn't notice 7 gig. That's nothing for them. But I doubt they normally get that much traffic to the ASM Atlas hosted data sets on their servers. So I started pulling down the whole thing as fast as I could. We actually downloaded it twice, which is interesting, because we wanted to make sure we had a 100% bit perfect copy. So we downloaded a second copy and made sure all the hashes matched. And yeah, I mean, that was the start of it. And then I'll talk more about this later. I want to let Niklas talk, but I also reached out to a few other people in the tech industry, and we've gotten open offers from a bunch of places to actually mirror this data set and provide resources for scientists to actually use this data set live. So I won't actually name names because I don't know how willing the company involved is to be named yet, but there's a company who in the next couple of weeks is actually spinning up a production IPFS cluster and Bakuyao cluster, and hosting a full copy of the data set with 50 terabytes of storage attached, all on enterprise grade stuff to host free computational resources for the ASM Atlas. So it's going to be incredible.
That is such an amazing rescue mission. That is, yeah, just that's brilliant. Niklas, tell us your side of this, or part in this as well.
Yeah, thank you. I mean, this is the, it's one of these stories where you don't know what you're getting into the moment when you first see it. So it's a normal morning in Berlin, Europe time. I open my Twitter X feed. I see that not only was the Microsoft AI research team for biology sort of laid off, that was sort of circulating the couple days before, but also there was a researcher showing that some of his API calls were timing out. And he was very worried that maybe not only was sort of the research group shutting down, but also the research that the research group has collected. Now, that was a moment where I got very concerned, and I sort of started creating a bit of a Notion memo where I said, okay, this is serious, people. Like, we need to like act on this. And then I was just sharing this Notion link in the Slack. I think I tried to abuse the nft.storage service in the beginning to like save some data. And then quickly.
I'm not saying we got some panicked calls from those guys, but
that's not going to work well. It's going to be very slow, and they're not going to be very happy if I completely basically swamp their NFT metadata binning service with protein structure data. So I posted it, and it was amazing because I then sort of continued my day. I felt like I pinged people. I felt like I told the world that this was serious. But then, I went on with my day, eventually checked my Slack, and I get a screenshot from Wings where it basically has like already the data almost downloaded. I'm like, I didn't understand at first. It was like, so awesome that you're DMing me. It's great that you're helping me. And it seems like, have you actually downloaded the data already? He's like, yeah, I've downloaded it already. It's already there. And I was like, that's insane. So while I was sort of not paying attention, you just went forward and you did it. And that is amazing, because I think if you hadn't had that level of agency, we don't know. Like, I don't know whether it would have happened, because I was able to point it out, but I was sort of just checking what were our resources, and we would not have had the resources and to like actually pull it off, and you did, which is great. So I think this is a huge testament to the community strength of like the Filecoin PL sort of extended network, where you're among company of people that have the sort of same values of like making sure that data is accessible and for many people. And this is, I think, a great story as a case in point.
Yeah, definitely. Yeah, sorry, go ahead.
Sorry. I was just gonna say it's there was kind of like an element of like us for forgiveness, not for permission with it. But I knew that like this sort of data set would just, everyone at PL would be in support. And when I brought up, hey, I started pulling down this data set, and we're using some spare infra that we had left over to go and like mirror this and make sure we have a copy of it that's safe. the response I got immediately was like, how can we help? Like, what resources do you guys need from us to make this work? So PL's been incredibly supportive of it, and we're excited to be part of it. We're excited to have IPFS involved. We're going to be archiving to Filecoin up to a bunch of different storage providers to keep the data like permanently safe as well. So we're going to be working with LabDAO and other groups to preserve this going into the future. But yeah, it was really cool, like the response that PL had to this, and we're really excited about it.
Yeah, this is the power of community too, right? Like you were saying, Niklas, and developer community especially. I think like there's not too many other communities that are this, oh, let's do something about it. Here you go. No, it's amazing. And can you tell us a little bit, for those that might not be familiar with this, maybe Niklas, you can tell us a little bit about what's contained in this data and what it's been used for in the past? Because I know I'm curious to know a little bit more about that.
Yeah, absolutely. So I created actually a couple slides. I can share my screen where we basically do a very speed run through, where is currently the sort of scientific debate in 2023 on, especially structural biology. Why is this data set so important? And just sort of to give everyone an update and also be fair to the Meta team, the data is actually still available and browsable. So I think there are people at Meta that have, sort of understood the importance of the ecosystem, and so at least they're delaying whatever they might do in terms of their restructuring. So the good news is people have noticed this. The Meta team has sort of kept some of the data online. There are other research groups that have also taken note. So sort of it's actually a testimony to the wider scientific community as well, that people are sort of seeing this as, okay, this is too important. We can't just shut this thing off. That said, IPFS, I think, is like such a good framework to like store these types of data, make sure that they're addressable, they can be peered and repinned. So I'm very, very glad that we have this resource, and I can share in just a moment why it is so important, and also sort of forward looking what I think we can do with that in general with this data set, but in particular also with the power of content addressing when we think about what this data set does and can help scientists with in the future.
Yeah, amazing. Feel free to share your screen as well, Niklas. You can share that at any time.
All right.
Happy to kick off your presentation as well if that's part of it, and you just want to roll right into that. We can do that now.
Totally, yeah. We certainly that sounds good to me. I'm just gonna, I divided it sort of in two parts. So the first part is really sort of this conversation. What is this metagenomic atlas and why is it so important for, I would go as far as to say, all of humanity? And then we can take a sort of a bit of a break and transition to, what is LabDAO anyway, and what are we doing? Why do we care?
Yeah, fantastic. Let's do it.
Amazing. Okay, so this what you're seeing right here is the metagenomic atlas, or it's basically 10 million at least data points within the catalog that spans 700 million proteins. So just, I think, to like establish some common groundwork, we're going to do a very speed run through the last almost 70 years of biology research. So in the 60s and sort of a bit earlier than the 60s, we figured out that DNA is sort of where all our information is stored in the body, and however all the actual work that is being done is mostly done by proteins. So proteins are these very complex 3D structures, and they can do all types of powerful things. They can catalyze reactions in your body. They can give your body structure. They make sure that neurons fire. They make sure that muscles move. So really, these are the engines of life. And if you think about, what is the building block of life, most of it is really, I think, proteins. Now, proteins are encoded by DNA. And while we were pretty good at understanding how DNA is structured, and we spent the last 20 to 30 years sequencing a lot of DNA, what we weren't really good at was understanding the laws of how proteins work and how DNA sequences map into protein structure. And understanding protein structure, however, is so important because, as I said, that's the actual building block. That's where actually the work is being done in biology. So if we understood the laws of how proteins fold and what determines protein structure, I think there will be a new unlock in progress, not only in biology, but in all of human manufacturing. Now, the exciting thing is we are exactly at that moment right now. And this resource plays a very sort of important role in all of it. So for the audience that's more into sort of data handling, I created a little small analogy that motivates some of the problem in biology and why this resource is so important. So you have this flow from DNA to protein, and there's actually a bit of compression. So protein structure is actually like a compression algorithm. So there are multiple DNA sequences that can lead to one protein sequence, and then multiple protein sequences can lead to one protein structure. So the protein structure is this like very dense compression of biological information. And if we really want to design biology, or we have a new organism that we don't know anything about, and we want to like learn about it as fast as possible. And by the way, there are so many organisms that we haven't understood yet. The fastest way to do that, the highest compression way that we can learn about biology, is by looking at how proteins are structured. Now, structuring proteins has been extremely hard, very, very hard. And people have already sequenced a lot of DNA and collected large databases of how these proteins look like, but predicting the structure was something that was impossible for a long time at a high accuracy. So this is a screenshot that describes one of the biggest protein sequence databases, or DNA sequence databases, that we have, which is the Magnified database. Now, what we saved basically was the Magnified database, but in a very interesting transformation. We didn't save the sequencing data, we saved the protein structure data, which is this like very efficient representation that was so hard to calculate in the past. But through some of the recent progress in just the last 48 months, and this is just one example for an algorithm that's called ESM that was developed by Facebook. This is basically a ChatGPT for proteins. So this thing is fed a lot of DNA, or more particular amino acid sequences, and it just tries to predict the next amino acid. And as almost like a byproduct, it learns how these proteins fold, which is like close to magic if you think about it for the first time. Now, what this Facebook team did, and other groups have done as well, is they used these models that have learned how to predict protein structures, and they applied them on this huge catalog. And the result is this. It's the largest collection of protein structures that we know for basically almost all the biology that we have characterized or sequenced so far as a society. I think that's, and just for perspective, our body, we have about 20,000 genes. So we have, in that order of magnitude number of proteins. These are 700 million proteins from different species, a lot of bacteria, fungi, mammals, insects, a lot of information that has been collected in this large corpus. And as I said, it is the most efficient compression of biological information. So if you have a new biological specimen and you want to learn about the function of a gene that you have never seen before, probably one of the best ways to learn about what that gene does, or what that organism does, is to go into this protein structure space with these new models that we have and look for a neighbor. Now, if you want to look for a neighbor and you want to look for references, you need to have a huge atlas of structures that are already available. And that atlas of structure was sort of, potentially threatened to be taken down. And it was extremely important that we create this reference of protein structures, for the I think the years to come, and make sure that it's properly backed up. And I'm very glad that, we got to this point, and the community sort of stepped in, and we moved this thing to a peer-to-peer storage system. I'm going to take a break here. Maybe there are some questions, or you're curious to sort of learn a bit more about why this is so important.
That's incredible, honestly. This is my brain's just blown at the moment. So
okay, well, I think that I can maybe just compress it to everyone's like, why is this so super important for our planet? Because if we have these data, if we're able to like predict protein structure, we're able to build better therapeutics to fight disease. We're able to engineer enzymes that can degrade, stuff that's polluting our planet and even completely avoid pollution because we come up with better materials that are biodegradable, for example, and instead of like polluting the planet, we can avoid the consumption of fossil fuels because we can synthesize alternatives. These things make me personally very excited. They make a lot of other people very excited, and I think we live through this very interesting period in history right now where we were sort of able to like not split the atom but fold the protein, and this was basically one of the largest collections of folded proteins, and it can serve as a very good sort of reference point if you want to do research in this regard, in this direction.
Yeah, this is amazing, and great job on saving it as well. I immediately thought about how much quicker we can get to things with some sort of protein LLM, which is what you're suggesting here. This is some sort of protein AI. This is the data set for a protein AI, and that's like, I can't imagine where that goes, but I can imagine there's a lot of good that can come out of that. So yeah. And I'm sure like there's a lot of scientists looking at this going, we could, do something with this. We could make it accessible to the world. We already have just about. This is available on IPFS already. So yeah, it's just incredible. I can't wait to see some of the things that come out of this as well. I'm sure there's been heaps of things come out of it already with Meta having it. Yeah. Have you got some examples? You're nodding your head there.
Yeah. So there's one example, which is, I think is a really good case study. Here's some tweets to document all that happened. Let me just maybe open actually the catalog. Maybe we just take.
Oh, right. Yeah. Awesome.
Let's do it. Let's see whether I can exit full screen mode safely. Oh. Can you still hear me?
Yep, we can.
All right. Well, my computer started to like do funny things again, and we already started 10 minutes late. So maybe we don't disturb it today.
It's fine. But we can always like carry over a little bit if we need to.
So basically, what you can do when you look at how this atlas looks like is there are areas that are underdiscovered, and I might just have to restart my browser to be able to show that to you. But let me just open a new window, and we are able to do this. I'm just going to open the ESM Atlas browser right now. Okay, so let me stop sharing my screen for a moment, and then we just open the Atlas ourselves. Okay, so let's do it. As I said, the Facebook team has not yet taken down the Atlas, which is, I think, good for everyone. And I don't know whether you can stop sharing my screen for me, whether you can turn basically off. Yeah. And then I'm going to try to share the screen again. All right, this is, I think, not working as much as I would like, but I'm going to share the link so everybody, you should definitely play around with this data, and sort of see it for yourself if you have a moment. We're going to put that in the show notes. There you go, exactly. So you can access this link. Thank you, Ali, for sharing your screen. This is great. So you can browse this, and let's just zoom into it. And you notice that there are different colors. So there are colors that are very light, and there are colors that are dark. When the color is dark, it means that we understand these proteins really well. And what does understanding means? It means we sequence this gene at some place in the world in some microorganism or plankton. And then we looked up in our existing databases, does this look like something we know? And when the dot is blue, it means, yeah, we were able to find something that looks very similar that we understand. But then when you look at this, there's also a lot of like very bright spots here. And the bright spots are proteins that we know very little about, that we have not a lot of reference information about what this, is there's a sequence that we know or is there a protein that looks very similar to it? So there's basically a lot of bright matter, a lot of material, a lot of protein information that we just don't yet fully understand what it does, or we haven't seen something like it before. And with these protein language models, with these structure prediction models, we're able now to like fold these sequences and actually look at how they look like, and hopefully we can learn something new from that. And even more importantly, if we sort of see something else like it in the future, we're able to like just put it on that map and have some reference points, and can very quickly learn something about how proteins are folded. So this is, for example, the entry that looks as similar is the closest looking entry that we know, and you see that they're not very similar, right? So there's a lot of protein. There are a lot of proteins in that catalog that we just don't know a lot about, and some of these proteins might help us identify a low carbon way to process certain materials, or they might help us identify new therapeutics. We don't know, but it's certainly a very important resource to keep studying. Yeah.
100%. This is incredible. And it is full of stars, Ben. You're absolutely right. That's originally what I thought. I'm like, this is a whole galaxy. And I guess it literally is, really. It's a whole galaxy of things that we don't know about, and on unknown science and unknown use cases, which is just fantastic.
Yeah.
Really cool site that I'd never heard of before. So this is awesome. And sorry, I'm probably making everyone dizzy zooming into this.
Do we know if like the rest of the ESM Atlas resources are open source, like this website and stuff? Because we could also mirror that.
Yeah, I actually don't know whether the website is open source. My current guess is that it isn't. And there are also other great tools that I wanted to share with you about, which the other tool that is available is there are now increasingly good search engines to search through 3D structure space. So you can obviously manual search it, but ideally you want to run queries that are really, really fast, because you have a lot of proteins in there. And one of these tools is called FoldSeek, and I think Ellie, you can probably pull that one up as well. So FoldSeek lets a researcher drop a protein structure that they found, and then basically look up, what are all the sort of through all the databases that we have, how proteins are look like, to find proteins that look similar sort of in 3D. And again, as I said, because 3D structure of a protein is like the biggest compression we know, it's a really, really efficient way to like look for, is there something that we've seen like it as well. So if we just, for example, submit this query, what Ellie's sort of scrolling through right now are the 3D coordinates of the protein, right? A protein is basically a graph in 3D. Now you see all the proteins that look like it, and you see that they have, when you click on it, they're all HTTP links, and they all have accessions. And this is, for example, the AlphaFold database. So the Google team is also maintaining something like this that is mirrored by the EBI, a nonprofit. So the EBI is sort of taking it on itself to make sure that these data are mirrored, and it's not on Google servers as far as I know, but really the EBI in Hingston is making sure that this data is available for good. Now, one thing that I'm very interested in, and we're now sort of peeking into what can we do with this data that's content addressed, is make sure that like a database search like FoldSeek is possible, and over maybe data that's currently available in hot storage, which I think is happening in FoldSeek under the hood. They're actually like, when they're running the search engine, they actually have a lot of this data locally to enable a fast search. But when you look at these, what would be really good, I think, would be to also have eventually like a version of this where there's additional column with the content ID. Like make sure that you can refer to this protein irrespective of its sort of accession, or irrespective of the HTTP link under which it is currently made available. And I think that's currently not yet the case, and in a way that I would like this sort of ecosystem to contribute to this line of research, to make sure that we have this information available, and available for good. And you can use some of these search engines that are made available, but when you search something, you don't only go and download it directly, but you also can access the content addressed web and refer to it with a very permanent identifier. So that's one direction that I see we can take. Now there's another direction that I think we can take, and this is, I think, the exciting part of why we're at the COD working group Live Jam, is because we can not only store data and share data using IPFS, we can also do computation. So what if we don't only make this data available, but we make sure that it keeps growing, that when a scientist is using some of these new fancy protein structure prediction models, and ESM is just one of them, that we make sure that these data are added to the mix, that you always work with basically the living graph of new protein structures that are being predicted. Now, why does this matter? This matters because of two reasons. First, predicting a protein structure from a sequence still takes a good amount of time and a good amount of GPU and electricity, so you don't necessarily want to waste the energy that you do. But also, the person that ran a query, they probably know something about that structure. So if you find a protein, or if you're interested in a protein that looks just like something that someone else has before predicted, they might be someone you should talk to, because they might know something about the problem you're trying to answer, because they worked on a very similar protein. Now, what we would need for that, however, is a way to dynamically load or add new data into such a mix whenever there's a computation run where we predict the structure. And this is where Bacalhau in particular comes in, and what we've been working on at LabDAO. Now here's the sort of a bit of note to the editor. I seem to be a bit frozen on my way to share the slide deck, so I might have to turn it off and turn it on again to see whether I can keep running the presentation and take you through that second part of the conversation. Is that okay?
Sounds good. Turn it off and on again. Let's do it.
I'll be right back.
Incredible work. And I might take this opportunity while Niklas is just rebooting himself to talk about the open data hack, which is on at the moment, which you might be able to do something with this data with, even, possibly. So Open Data Hack is an encode Filecoin hackathon that's running at the moment. There's a bunch of bounties going on. I think you can still join. It's open until September 27th. So submissions are due on September 27th. So you could, of course, join up and do something with this LabDAO data that's been, this ESM protein data that's been saved, if that's of interest to you, or anything else. We also have a LilyPad bounty. So LilyPad is the project I'm currently working on building. We are building a trustless compute network based on Bacalhau initially, but incentivized Bacalhau. So basically, kind of like Filecoin is a persistence layer for IPFS, LilyPad is kind of an incentivized layer for Bacalhau, kind of giving this trustless compute network availability. So, and we're definitely talking with LabDAO about how we could potentially integrate with them in the future as well. So currently we are just doing AI inference modules, although someone's just contributed a DuckDB module, which was an amazing feat for the hackathon. They've put in a pull request, so as soon as we reboot LilyPad, that'll be live. You'll be able to use DuckDB on LilyPad, which is pretty awesome as well. Welcome back, Niklas. How'd you go with your screen there?
Well, we'll see it in a moment. Let's
people about the open data hack that's on at the moment. They could possibly do something with that protein data that you shared with us as well.
Oh, amazing. Yeah, that would be great. Okay, so I think you should be able to see.
Yes, I can see your screen. Here we go. Swapping now.
Okay, so we're at LabDAO, we're part of a group of projects that we came up, I guess, with this name of decentralized science. And what we're really interested in is new ways to distribute scientific results, but also make sure that there are new mechanisms to raise funding and use some of the latest tools that Web3 has to offer for scientists all over the world to come together and collaborate. And we're not alone. There are a lot of other projects in this space, and it's been very excited to see it grow so fast. And one thing that we're really interested in is, making sure that scientific results are reproducible, and that when scientists do work that there's assignability to like what a scientist did. And in right now, and probably for the last 500 years, the way that scientists have internally given each other credit was by citing papers and by counting how much is someone citing a paper. And that was sort of their version of like creating a record or a permanent record of the work that they did. But the technologies that we have today, I think, give us two things. First, they give us a better way to refer to like a publication, because we have actually like fully content-based ways to address information. And also, maybe the atomic unit of science is not anymore a fully blown paper, but just a computation or a new data set that was added. So what we're really interested in is making sure that something as simple as contributing a data set or running a computation goes into the scientific record and can also be referenced, can be owned, and that's really what we've been working on. And under the hood, all what we're using is we store data on IPFS, we compute over it using Bacalhau, and then we have a very lightweight non-fungible token contract over it that allows us to always make sure that when someone is using one of our computational biology tools, that they have a unique reference to, okay, I did this work and this was my result, and this was the data from which I generated my result from. Now, there are a lot of problems with storage in academia right now. So there's link rot. So for example, if you looked at this paper and you clicked on the link, you're actually not going to be successful because that link is just gone. And whatever resource was shared there. So in this particular case, it's an algorithm that did calculations over certain biological components. That is just not recoverable in a permissionless way. Surely you can probably still contact the scientist, but you're going to have a hard time just doing this on your own. And basically the TLDR is that most scientists are not great archivists. So we need to build products that make the archiving a byproduct of the work. You just want to do science, and the product just under the hood makes sure that whatever you're doing, if you want to, is recorded publicly. And Bacalhau is just like a perfect tool, to like power computation in biology, because in biology you face large data sets and a lot of prototype grade code with exotic dependencies. So you need to make sure that A, you do the computation where the data is stored, and then B, you containerize these applications because they all have very, very easy to break sort of dependency stacks. So what we did was we created a very simple sort of JSON model to define tools for scientific computation. It's very heavily inspired by the Common Workflow Language Standard, CODwg. We sort of sometimes internally joke that our riff on it is called IPWL. It's the Interplanetary Workflow Language. It's a simple JSON object that allows you to like define the inputs of a computation and the outputs, and a container image that with an argument that does all the work for you. And we have some sort of approaches that we take when we think about what information should the record track. It tracks both the inputs that go into the rest into like a scientific product, what is actually happening under the hood, and then the output. So in this particular case, example, we have we want to bake a pie, a lemon meringue pie, and we have 5 different inputs. We have a model, some computational sequence that is processing these inputs, and then we have an output. And this translates not only to creating cakes, but also to doing computational biology or drug discovery research, where here we have a protein and a small molecule, for example, as an input, and we want to try to predict how this drug-like molecule interacts with the protein. So we use a model, can be a machine learning model, can be a sort of more classic algorithm to predict the interaction, and then we post the result to IPFS. And how that all looks at the end is it's one token. You can look at it at OpenSea if you want, and the metadata of the token basically contains this small computational graph where we have the inputs here, the tool that was used and its reference here, and the outputs over here. And we can attach licenses to these contracts. So for example, one contract that we currently have on Optimus and Testnet gives the owner of the token the exclusive data ownership rights over the result, which we hope creates a bit of a way for scientists to like own their result and make it easier for them to collaborate across the globe, where they're not necessarily slowed down by national sort of copyright or institutional copyright laws. Now, what can you do right now is you can do some small molecule binding, but you can also do protein folding, which gets us all the way back to the ESM atlas and why we're here today. There are a lot of other tools that have onboarded, and I invite all of you to sort of take a look and check out the website to explore what's available. Now, we could do an interactive demo, but I might just want to pause here and just give it a perspective for what I think we can do now that we have the ESM atlas and tools that allow us to basically fold proteins and publish them to IPFS as well. Which is that when we create like this data that was the ESM atlas on IPFS, we basically had a static snapshot of all the protein structures that had been calculated up to this point, or a lot of them. Now, what I think could be very exciting is if with our tools and powered by Bacalhau, you predict protein structures, and you might not only predict them using the current tools, but in a couple of months there might be an even better tool that comes out. You use that tool on Bacalhau, and your result is basically another contribution to this pool of protein structure data that we have as a sort of global community have collected. And what I'm really interested in is building a way to like make all of that data visible, not only the data we collected a couple days ago, but also the data that is going to still arrive in the years to come, where scientists all over the world like try to predict structures based on amino acid sequences. Now, what do we need to do in order to get there? So first, on the technical level, we need to make able that we can index these new predictions that are made as people are using the tools on Bacalhau. So we need to make sure that we have a catalog of not only the existing data that was currently pinned, but also from the new predictions as they're being added. And then, we, I think, need to all aggregate it together and use some of these search methods where we say, okay, for every protein in this ESM atlas, what is the content ID? So we can have a lookup table, and we can sort of enrich tools like FoldSeek. So in the future, you don't only have the target and the HTTP link, but you also perhaps have a content ID column in here where people can actually refer to the protein based on its structure and its unique identifier in the interplanetary file system. I'm going to take a break here. Very happy to like answer sort of any questions on how we sort of designed the system where we want to take it. And then I have sort of a little demo where we actually run a notebook ourselves to get everybody sort of a feeling. And at the end, there's also a link that you can just go to here. These are our tutorials. So you can run 2 different tutorials, and there's going to be a 3rd tutorial just added in a couple of days as well.
No questions at the moment, Niklas. So I'm keen to see the demo if that's cool with you.
Of course, yeah. Let's do it.
And
okay. Go ahead.
Labdow.xyz, docs.labdow.xyz, did you say it was that?
Exactly. So you can go to docs.labdow.xyz, and then in the tutorial sections, you can see some notebooks that we're maintaining.
Awesome.
In the docs, we'll also like walk through some concepts and how to install it if you want to run it yourself. We have currently some Python bindings. It's a very like small package, but it gets the job done. All right, so we're just opening the notebook. So what we're doing here is we wanted to basically meet scientists where they usually do their work, which is they use Jupyter notebooks, they use Python notebooks to do most of their work. It's very interactive computing because you're not building products, you're exploring data and you're analyzing it. So we needed to build something, needed to meet them where they are, and build an interface to peer-to-peer compute and storage to do that. So what we're doing now is we're going to be folding a protein, and this is also based on the Colab Fold notebook. So a huge shout out to Sergei Ovnikov and Martin Steinegger, who've just been amazing contributors to this community. They built a lot of the tools that everybody is using to get their work done right now in this space. Okay, so what we're going to do here is we can specify a protein structure that we want to predict. So come up with an arbitrary sequence. If you want to, we can place an Easter egg in here. So we can even say COD work. We can do COD. There might be some error because the amino acid alphabet doesn't have all the letters in the alphabet, but C and D should work. So let's just say C and D are enough. We give our tool our job a name, and then I give it just an Ethereum virtual machine wallet. So this is our LabDAO default wallet. So we're defining these parameters, and then what we're doing is we're folding the protein. So what's happening under the hood are 3 steps. Our current package basically just knows 3 functions. The first one is, we call it init. So what init does, we can just run this in a stepwise way. What init does is the initialization basically creates the computational graph. This is basically just a JSON document. So we're referring the tool configuration file, this CWL spinoff that I mentioned in the presentation before, and we just point to a data file. What's happening under the hood is that during the initiation step, this data file, which is currently local, so here you see this file here, this data file is going to be pinned to IPFS to a node that we are running, and the content address is going to be put back, and we basically then have all we need in terms of the computational graph that we need to execute. And then the next step, we run an operation that we call plex run. So we submit that, basically DAG to our requester node that we're running. So everybody that's more familiar with Bacalhau, there are 2 types of nodes: a requester node that basically sits on top of the network, and there are compute nodes that actually do the work. So we send that to our requester node that we're running, and it's going to pass it on to the compute node. So the work's going to be done. And then last but not least, the third step is we get the result back. We now have our input data with its content address on IPFS. We have all the information about the tool that was used, and we have our output data. So we have our triplet complete of input, function, output, and we're going to mint that as an NFT and send that to the wallet to make sure that we just created a permanent record of someone did this computation on the distributed web. So now let's run this thing. So we're initializing this thing. We're pinning our what we currently call the DAG, our compute instruction, to the permaweb here. And then it's locally creating a bit of a job directory so it can keep track of all the files that are going to be produced. And we're submitting a job to the Bacalhau network. So let's do something fun and actually look at our IPFS gateway and check out what did we actually just pin to IPFS. So this is basically what we currently call the the DAG. So we said, okay, we created a job by the user. This is the wallet. There was not yet a Bacalhau job ID at the time that this was created. So it's basically the first thing. And the inputs is a file that's a FASTA file. We can retrieve that thing too if we would like to. And the tool that's referenced, has a content address here, and it's called collabfold. So the outputs are still empty because we're still waiting for the Bacalhau network to give us the outputs, right? So we got to be a bit more patient. In the meantime, however, one thing we can do is we can, let's check out what else is referenced here. So we can take a look at the files. Start with this input file here. Now this is our FASTA file, the protein sequence that we wanted to predict, and over here we have our little Easter egg for the COD working group. Now we can go back and we can take a look at our tool configuration file, and this looks a lot like the example I showed you before. So it's just plain JSON. It has a name. You get to sort of know what it does. You have a container entry point. You have a container reference, some hardware-related flags that the scientist that developed the tool tells the community you should run this with a GPU and you should have networking on. And then it says, okay, I expect as inputs a protein sequence, and there's one additional parameter that we don't need to explain now. And as an output, I'm going to give you a protein structure, and this is always in PDB format for all the biologists that are watching this. I'm going to call that output. I'm going to type that as a file that is called best folded protein. And then I'm going to create a second type, which is called all folded proteins. It's going to be an array of files that they're going to have ranks. All right, so this is our deep dive. Now, this is what we submitted to the backlog cluster just a moment ago. So let's see, how things are looking like at this point in time. Okay, so this thing is still working, but in the meantime, we're already seeing this message over here. This is basically our IPFS, our libp2p telling us, I received the data. I'm actually downloading it into the session. It's obviously a bit inefficient. In the future, we're just going to keep the data on IPFS. We're not going to download it back again into our local workspace. And now, we're actually putting our contract that's on Optimism testnet right now, and we're minting a token. So we can actually take a look at that token now. And that's, I think, pretty cool as well. So sort of closing off this demo with going to OpenSea. We have this unidentified contract, which is currently our testnet contract. And let's see whether OpenSea can be a bit slow at times, but let's just see whether we have some information here on the
I gotta say, I like the sound of anisotropic Heisenberg was one of those.
Yeah. So we have a mechanical Dirac neutron, genetic Rubin centrioles. There's a token for everybody. We're not. I don't think I saw like what we had, what we just created right there. Sometimes it has a bit of latency. But let's take a look at isotopic Tesla antibottom quark. And we just take a look at the token ID. It's a bit ugly in terms of its rendering right now, but this is you'd have to believe me. This is a JSON document that basically just summarizes everything I just showed you as individual pieces in one big model. That what we believe is going to be like a permanent record where we say, okay, this is a research object that's all scientists welcome. Now everybody can access this data. And the tool is also fully reproducible. So you can not only like reproduce this thing, you can also remix it, change some of the parameters, see what happens. And what we now really want to do is we want to make sure that this result here, although it now entered sort of the record because there's a verified entry, we want to do something more. And I think that's what this collaboration around the ESM Atlas stands for, is we want to make sure that this data is not only sort of registered, but also queryable. So one thing that we're really interested right now is building an interface and making sure that we index these results that people create, and we feed them back into sort of search engines that the scientific community is currently using. So they start seeing, okay, there's actually some utility here when I use tools on the content addressable web. All my scientific colleagues can sort of see and contribute. At the same time, I feel safe that I'm sort of not getting scooped. Some scientists really care about not that like sense of ownership, because you actually have that artifact that is very much yours, that you were the one that did that computation or came up with that idea. And sort of with that, I think that is currently all I have. We'll keep onboarding more tools, try to make the UX better. And we really, we're really excited about the support we've got from the backlog community in particular, and the sort of wider Protocol Labs, Filecoin, COD community in general. Thank you.
That was amazing. Thanks so much, Niklas.
I'm now.
I just wanted to briefly touch on, sort of the ownership and what it actually means for scientists, that, moving on, all this new data is going to be generated by them, but also owned by them, and it can be completely tracked. Like, what does it mean actually for funding and the scientific, landscape?
Yeah, so the way that currently scientific organizations look like, they're a bit like record labels. So you as a scientist work for a university or an institute, and the institute, depending on the region of the world in which you're in, There are some exceptions. The institute usually has rights to all the data that you generate and the inventions that you make, and that oftentimes is a good deal, but unfortunately, more often than not, it's not a great deal for the scientist, for the inventor. So what this enables you, if you're an independent scientist, or also if you're a scientist right now, you can keep track of everything you produce, and we use sort of it's a license called CODwg license by A16Z that gives the owner of this NFT like the full rights to the data. Certainly, when you're a scientist in a traditional organization, they will still say, well, this is all fun and games that you have with your NFT here. We still have this like contract with you that you signed that all the data is actually ours. But we hope to sort of start a change in which sort of scientists get more sort of ownership over their output, the same way that sort of artists went direct in the last years. I really want to see scientists go direct in the next coming years and build their own audiences, make sure they create collectibles, make sure that they have control over their data, and they can raise the funding into sort of collectives of data where they steer and have control over how that information is used. So I think it sort of just creates basically a token that scientists can start to collect and sort of shift maybe some of the negotiation balance during the funding process to make sure, okay, I actually own my results, and I can sort of have a bit more freedom in the way that I seek funding.
Amazing, so powerful. It's really going to change that whole landscape. But I think also in terms of what type, research is actually getting funded, because all of a sudden they can actually get going, create some of the results really fast, and then showcase that, to have, they actually have a choice,
Yeah, thank you. I think there is something here just about like the sort of radical, almost like barbell strategy where you as a scientist, you own the rights to the data, but at the same time, you share them the most possibly open way because you actually want people, like you want the distribution, you want people to use your data and like find it interesting, build upon it. At the same time, you have that sort of, I guess, sense of authorship that is very important for scientists and the scientific community.
Amazing. Well, thank you so much for sharing all that with us, Niklas. We really appreciate it. And you, Ben, for joining us as well. Great.
Thank you for having us.
Yeah, it was really informative. I've learned heaps tonight. And yeah, can't wait to see how this moves forward and to work with you as well, Niklas, because I know we're talking about doing a collab with Lilypad. So excited to be doing that in the future as well, and what we can dream up, like some powerful stuff happening here. So thank you so much for this project and what you're doing. And anyone else out there that's interested, hit Niklas up. He's in the Backyard Slack and in the Filecoin Slack. I'm sure he'd be happy to take your questions. Also on Twitter at LabDAO. So yeah, you can find him at all those platforms, and check out the docs as well. Really great docs. Thank you so much, everyone, for joining us tonight, and we'll be back again soon in the next couple of weeks. Appreciate you joining us, and have a great weekend.
Thank you.